So I'm, as Carol mentioned, mainly working with the phenomenon of parahydrogen, and I'm also interested in this related phenomenon of nuclear spin singlet states. Per se, my group doesn't necessarily work at zero or ultra low field at the moment, but I had a great opportunity within the ITN, within the ZULF collaboration, to actually do some zero field NMR, especially together with Szymon Pusztelny. And on the other side, Göttingen is a very interesting place for me, where I can also apply all my techniques to very high magnetic fields. So especially like to mention this, since this week, we actually have a 1.2 gigahertz magnet who was brought up to field. And what I'm going to show you is some data from my group that actually spans various orders of magnitudes in magnetic field, ranging from 20 Tesla down to actually zero field NMR. And NMR is the reason why we're all here, I guess. It's a very wonderful technique, very versatile, that has been used for many applications in, for example, metabolomics, structure analysis, various in vivo applications. And I find it very interesting that we can even measure dynamic phenomena such as diffusion, and we can even do this in vivo. But as this is the hyperpolarization session, I'd like to show a few of these slides, explaining a bit more again what we're doing. As we know that magnetic resonance is very insensitive intrinsically. So I picked up the values from actually the 28 Tesla magnet, which corresponds to this 1.2 gigahertz. If you put 1 million protons into this magnetic field, then your spin polarization is actually something like 80 ppm, right? So since we're just looking at magnetization difference or orientation differences, we can only get 80 ppm, shown by this particular red spin over here, that contributes to our net magnetization. And since if we can compare the potential of hyperpolarization to the highest available magnetic fields, then at least for protons, we can still get a signal enhancement of over 10,000 fold. And if you do the math, this will speed up measurement times over many orders of magnitudes on one side. But with these large enhancements, you can also do very nice, interesting new experiments. And I think what got me pretty interested, that by the year 2013, this was about when I finished my PhD, this interesting paper came up where people started polarizing pyruvate, and they showed that you can actually inject it in vivo into humans and differentiate between cancer and prostate cancer, about here, and show that from the hyperpolarized metabolism from pyruvate into lactate, that the cancer is actually metabolizes differently than the normal healthy tissue, where we only see this single peak of pyruvate over here. So very nice and interesting work, and this was done with the current state-of-the-art methodology, dynamic nuclear polarization, which is a great technique if you want to hyperpolarize tracers in batches. And what got me really interested in was then the phenomenon of parahydrogen, because parahydrogen allows you to create polarization very quickly, and somehow has the promise that you eventually can produce continuously very highly polarized samples. So parahydrogen, the spin isomer, was originally proven to exist by Carl Friedrich Bonhoeffer, which you see on the right side here. Something like over 90 years ago. And we'll see that hydrogen actually has these two spin isomers, orthohydrogen and parahydrogen. And you can specifically enrich this para state to about 100% at 25 Kelvin. And then even if you take away a catalyst that is necessary to do this ortho-para conversion, then you can heat up the parahydrogen to room temperature and can store it even over weeks. So that was something done like 90 years ago. And then, I would say over 50 years later, some very smart people figured out that you can actually use this phenomenon also to be useful for NMR, because you have a type of spin order. And what you can do is you can take an unsaturated bond over here, you can apply a catalyst and hydrogenate your unsaturated bond, and the singlet spin order in this hydrogenation process somewhat broken, and you obtain a hyperpolarized molecule on this side. So since my talk was then tailored a little bit in metabolites, I would like to show you then this interesting technique that came up some 5 years ago, introduced by Francesca Heinrich and Silvio Eimer, where they actually made use of this hydrogenation reaction, and you have your hyperpolarized proton order, and you then somehow transfer the spin order to a heteronucleus, which is inside the metabolite. You do a very fast cleavage of the side chain, and then in the end you obtain your hyperpolarized metabolite via parahydrogen that you can then actually use for metabolic analysis. And yeah, that's some procedure that just takes a couple of seconds to accomplish. And yeah, my group then started to look into possibilities of really driving this polarization to its maximum. And we started doing experiments at 7 Tesla magnetic fields with this deuterated vinyl acetate, which you see here. It's commercially available completely in the deuterated form without carbon-13. And that has turned somewhat into our guinea pig molecule to do these metabolite studies. So all the data I'm showing you here is basically carbon-13 in natural abundance. So as before, what you can do, you hyperpolarize the protons over here. Then you do a polarization transfer from these protons to the carbon. And in the end, you cleave off this molecule. And in the past years, we have worked on developing some pulse NMR experiment. So here's a sequence that we came up with that works in this particular high field case where we assume 2 spin order of the 2 former hydrogen molecules. And we're basically converting this 2 spin order then in the end into in-phase magnetization of this carbon-13 nucleus. And these were our, say, first experiments with this metabolite, where we worked with a little less than 1 millimolar concentrations of acetate, again in natural abundance, and we achieved this impressive polarization values of 60%. So for me, that was also a point where I said, okay, we are actually able to achieve very, very high levels with polarization with parahydrogen. Let's carry on with this and let's see how far we can actually drive this. Now you see we've worked with very, very low concentrations, so a bit lower than 1 millimolar. And the reason for this you see here. So I show you a spectrum with about a little bit more than 1 millimolar concentration. So what we're seeing here is we start having radiation damping effects. So this is 2.5 millimolar concentration. So our spin polarization in these 7 Tesla magnets all of a sudden becomes so large that you have severe interaction with the with your coils. And then all of a sudden our experiment falls apart. So we only achieve a very inefficient transfer to carbon-13 if we want to go to higher polarizations, such as 50 millimolar. 50 millimolar, that's like a concentration what people in the in vivo community typically aim for to run the in vivo studies. So then we started tweaking a little bit our sequence to fight these effects, and that mainly worked by introducing some gradients. So you see 2 realizations of the sequence where we actually have to switch on and off bipolar gradients to actually fight this large magnetization. So we're dephasing and rephasing it all the time while we run through the pulse sequence. Or we do a echo refocusing because we might have then convection or diffusion effects on top. And yeah, with these echoes and a constant pulse field gradient that actually broadens the line width of our NMR, we can actually get rid of the effects that we've observed here. And when we do the readout, then we finally managed to get very close to the 60% polarization in very highly polarized samples of this concentrated samples of the 50 millimolar. So compared to 7 Tesla, this corresponds to something like 100,000-fold signal enhancement. And we're on the way currently to make this happen for other metabolites like lactate, pyruvate, and amino acids. Okay, so now if we take this spectrum, or if we take this molecule, this polarized molecule, that's the same spectrum down here, We can show that we can cleave it. So that's a procedure that we haven't fairly optimized at the moment, since we're working on the way of achieving higher and higher polarization for different molecules. But if we do a 40 seconds cleavage, we still end up with 17% carbon-13 for the acetate peak, which you see here, which we could then use for some biological applications. So this was done at 7 Tesla. Now we're going a little bit lower in field because so last year we teamed up with Bernhard Blümich, who is famous for low field NMR, and we worked with him at in 1 Tesla spectrometers, and we could show that even in these benchtop devices we can create similar amounts of polarization, like 35% polarization, only within a few seconds in such portable devices. And yeah, this is an interesting molecule, and what we learned in the process that we were still or mostly struggling a little bit of creating a similar molecule with pyruvate. So vinyl pyruvate instead of vinyl acetate would be a very nice molecule. When it's completely deuterated, we would expect to achieve similar levels of polarization. So I see this one of the main goals in developing precursor molecules. But on this way, we actually worked together with Malcolm Levitt and Laurie Nass and developed some nice sequences for polarizing pyruvate. So now is the time, actually, if you want to look at the poster, please take out your phones and scan Laurie Nass' QR code here, which will immediately give you a link, and take a look at the poster in a little while after the talk. And I will walk you very, very briefly through Laurie Nass' sequence that he actually developed during a secondment in my lab in Göttingen. We start again with parahydrogen, and we have polarized parahydrogen here, and we're trying to relay this over an extra proton. Here you see this extra proton, and then from this proton, which is then polarized, we're trying to jump over to the carbon in this pyruvate molecule over here, resulting then in phase magnetization on carbon-13 polarized pyruvate. And this actually seems to work quite nicely. What we can accomplish here is we start with about 15% proton polarization on the hydrogen, and jumping over this proton to the carbon, we achieve about 7.1% polarization. That's about this spectrum over here, and you see the thermopolarized spectrum at the bottom of actually the precursor molecule before we start hydrogenating with 520 scans. Yeah, so please check out the poster, just scan the QR code and talk to him if you want to know more about this. And especially he working on implementations for ZULF NMR for these kind of molecular systems, which you will also see on that poster. Another interesting story about parahydrogen is, of course, the catalyst. For the hydrogenation reaction, you need catalysts. And this goes a little bit way back to my postdoc time, where I started to be interested in nanoparticles. And I produced these nanoparticles that are soluble in water, and they have certain ligand structures on the outside. And these kind of particles actually help us to hyperpolarize certain molecules in water. Here at the top, you see hydroxyethyl propionate, which was a guinea pig molecule that many people used in the parahydrogen society community at this point to hyperpolarize, because it had been used by Goldman and so on a few years back for actually in vivo angiography experiments. And the overall aim here was to make these particles that at some point we come up with a heterogeneous catalyst that you can eventually just separate easily from your solution. But as I will show you in a bit, there's more to it than just having a catalyst that you can easily separate. So these were my first steps. We had something like 0.25% polarization on these platinum particles. And the idea that we have why this actually works is that we require really these ligands to maintain the parahydrogen spin order. So let's say you have a bare particle which is several nanometers in size or even bigger, then parahydrogen can absorb to the surface, diffuse around, and we're losing the spin correlation. So that means usually we're running reactions at somewhat higher temperatures than 25 Kelvin. So the favored state is actually the orthoparahydrogen state, which is then quickly created. And we're not observing very much polarization if we use these kind of catalysts. With this ligands, apparently the spin order can be maintained of this parahydrogen on the surface. And if we then run our hydrogenation reactions, we see hyperpolarized signals. Going on a few years later, we actually now working with these rhodium catalysts that are kept with cysteine, so much smaller ligands on the surface that seem to hinder much more this diffusion of parahydrogen. And we're actually getting with this acetate over here to about 4% and 1 millimolar concentration. So we're on a good way on this, and there's still some work in progress. So now in comparing NMR, T1, and specifically of molecules to other techniques like positron emission tomography, which is basically gold standard in also clinical research, we see that these radioactive tracers have a half-life time of more than 20 minutes. Right? If you look at the 11C radioisotope, if you go to fluorine isotopes, then you're even getting longer in these times. And if we compare this to the T1 of pyruvate, and then look at some in vivo data that some people have collected at some point, you see that you can follow this pyruvate about 2 minutes, and then you don't see it anymore. So I'm very much interested in working on the possibility to actually prolong this traceabilities, to actually have a potential to trace these molecules, hyperpolarized molecules, longer in an organism. And then we found, first of all, this wonderful molecule, which is a quaternary nitrogen, and which you see down here. And specifically, the development of these nanoparticles that we had before helped us to achieve very, very high levels of polarization on this, about 12%. With the homogeneous catalysts that we tried, they seem to unfavorably interact with these kind of molecules, and we achieve 1 order of less magnitude on polarization with them. And a very interesting part here is that the T1 of these particular 15N nuclei is over 20 minutes, so you can really generate very nicely hyperpolarized signals, and you can still observe them via this long T1 after actually 1 hour of time. So as I said, these catalysts actually allowed us to go in there, and I think in the also recent years you saw in the FIP and SABRE community how important the adjustment of the catalyst is. I mean, we've seen catalysts that came around to do a transhydrogenation, where you see a lot of beautiful work to hyperpolarize fumarate these days, and especially to push to very high polarization levels on 15N with the SABRE technology. The invention of new catalysts was necessary. So it's very interesting, and I think pushing this catalyst science, we're able to access many, many more molecules that we can actually hyperpolarize very, very nicely. Another molecule that we got interested in, which we discovered, is this pyridinium species. So this pyridinium is interesting because it's a molecule that appears in many drug molecules, in many fluorophores. It's being used actually as drug delivery systems. And in particular, this 15N, the T1 in water is very, very long if you go to low magnetic fields. Low meaning we tested this to 10 to 100 millitesla. So we have a T1 of 8 minutes in these fields, and using the same catalyst you've seen before, we achieve very nice polarization levels over here. So I think from a chemistry perspective, this is right now my favorite molecule, and we're trying to design very interesting probes based on this structure. And this is a point where I would like to actually show you some zero field NMR data, because as I mentioned, we're working with Szymon in Krakow, and we've recently sent him a protonated analog for some thermally polarized experiments, and Shema just sent me this nice spectrum this morning. So you see this methyl pyridinium over here. And you actually see that there are some ZULF NMR going on. We haven't quite understood what all the couplings is and haven't simulated the spectrum, so this is very first data we're seeing. One interesting fact, especially about this methyl pyridinium, this is actually a molecule that most of you probably consume every day when you drink your cup of coffee. So this is some metabolite actually found in coffee. Yes, so in the end, I would like to show a few slides going a bit away from this T1 phenomenon to the so-called singlet states, the phenomenon which was basically introduced like 15, 16 years ago from the group of Malcolm Levitt in Southampton. But the idea is that, okay, we have these triplet states of two adjacent spin 1/2 nuclei. It's basically similar to what we have in parahydrogen, but these nuclei can be anywhere in any type of molecules, like 2 protons and these amino acids, for example. So in a triplet state, we're actually able to acquire our NMR signal over here. But then we can apply some very nice tricks to bring these two protons or 2 spin 1/2 nuclei in this singlet state, which have no NMR signal. And the feature is that very often this equilibration time from the singlet state back into the triplet state can be very, very, very long. So this is a very interesting method for potentially storing hyperpolarization for very, very long times. And often we can store this polarization, particularly at low magnetic fields, for an extended period of time. So yeah, I hope we'll hear a lot in the following talk from Malcolm about this. And what I would like to show you is some examples from my group. We've been very much interested for a year now to push this into in vivo application. And in order to do this, we thought, okay, first of all, we need to develop a NMR experiment that is very robust and that we can use over several spin couplings, spin coupling regimes, weak coupling regime, strong coupling regime. And especially the sequence needs to be robust, because if you think about in vivo applications, your organism breathes, and while the organism breathes that you want to look at, your NMR line can actually move over several hertz. So you need to be very robust in picking up the signal again once you've populated a singlet state. And so here's some results. We tested this actually on some A-beta 40, which is a peptide that you see over here that tends to aggregate during Alzheimer's disease. And this one has 6 different glycine residues. And you see very nicely, we have like a strongly coupled glycine. We have like a more weakly coupled glycine over here and several parts in between. And we managed to come up with a sequence that addresses one after each other all these different glycine residues, which is offset independent. And we thought, okay, nice, we have a sequence that we might be up to do some very interesting biology experiments with. So what we did first then is we actually worked on some ex vivo brain tissue that you see over here. And you can see that we have an NMR signal of the metabolites in the brain that you'll find over here. And by applying these singlet techniques, you can actually have a filter for specific metabolites. So out of these, I would say, wood of signals, we're actually able to filter out this particular glutamine signal by our singlet states. Interestingly, even in brain tissue, we found that the singlet state can be long-lived, which you see here. So T1 is 1.2 seconds of these 2 protons we see here. And the singlet state is several orders longer over here. We thought, okay, this looks very promising. Now we want to really try to push and get this into in vivo applications. And what we did quite recently is then we turned our sequence into a localized singlet filtered NMR experiments. And just this Tuesday, we had a paper published online where we actually can show first singlet filtered in vivo data in the hippocampus of a mouse. So this is the proton image of the mouse brain. And this red box is a voxel of about 42 microliters. And that's the hippocampus actually of this mouse. Here again, you see the nice wood of signals. And by applying this filtering technique, we're actually able to suppress all other resonances than the glutamate signal, which we were interested at the time, and can use this as a filtering technique. That was for me also like very first steps into in vivo imaging, and we were happy to have some nice collaborators around Jürgen Hennig in Freiburg who implemented the sequence on their MRI scanners. There's, yeah, I would say a lot technological development now to do, but otherwise I think it's very exciting because we're opening now the possibility to investigate singlet lifetimes even in vivo applications and see what we can learn from this. Yeah, so with this I would like to conclude and summarize that we've seen we can create very high levels of polarization with parahydrogen. We can store this hyperpolarization for tens of minutes in quaternary 15N nuclei. I've mentioned catalyst and precursor development is a very crucial aspect for parahydrogen to get more and more molecular probes to be used potentially for molecular imaging. And I've shown you that most recent results show that we can actually populate singlet states in endogenous metabolites in vivo. And yeah, we're looking very much forward to explore all these things further in the future. With this, I would like to thank my group that you see here, my funders, and my ever-growing number of collaborators. And yeah, thank you very much for your attention.